Hi everyone, let's compare and contrast some terminology related to genetic variations. I know how frustrating it can be to learn about these in class, so I decided to make this video to quickly go over some of the most confusing terms and directly compare them. If you want to learn about more terminology, or you want more information on some of the terms that I talk about here, you can watch my other video where I go over them more in depth. First off, structural variations, or SVs, versus copy number variations, or CNVs. Structural variations are genetic changes 50 base pairs or larger that are related to structure. So, inversions, duplications, anything that changes the structure of a chromosome would be considered a structural variant. Copy number variations, on the other hand, are regions of DNA 1,000 base pairs or larger which differ in the number of copies present in each person. Person A might have 3 copies of the same region, while person B might have 0 copies. Copy number variations can and usually are also structural variations. Next, structural variations versus single nucleotide variations, or SNVs. The abbreviations might be extremely similar, but don't get them confused. Structural variations are large genetic changes, while SNVs, as the name suggests, are only variations of a single nucleotide. These include missense, nonsense, and silent point mutations. Single nucleotide variations versus single nucleotide polymorphisms, or SNPs. Again, very similar names. When a particular single nucleotide variation is present in 1% or more of the general population, they get promoted to the term single nucleotide polymorphism. So, SNPs are the more common SNVs of a population. SNVs are also very commonly confused with indels. SNVs are usually referring to base substitutions. For example, a thymine might have been changed into a guanine. Indels, on the other hand, are insertions or deletions. While they can be 1 base pair long, they can also go up in size to 1,000 base pairs. Another confusing pair is variable number tandem repeats, VNTRs, and short tandem repeats, STRs, also called microsatellites. These are both referring to repeating regions of DNA that lie side by side or in tandem. However, VNTRs are repeating regions ranging from 10 to 100 base pairs, while STRs range from 2 to 7 base pairs. Aneuploidy versus polyploidy. Aneuploidy usually refers to the gain or loss of chromosomes. So rather than the wild-type 46 chromosomes, people with aneuploidy, such as those with Down syndrome or Turner syndrome, will have either 1 more chromosome or 1 less chromosome. These can be represented with 2n+1 or 2n-1 notations. Polyploidy, on the other hand, refers to the gain of entire chromosome sets. So rather than a haploid cell or a diploid cell, a cell can be triploid or tetraploid. For example, if a human cell were tetraploid, it would have 92 chromosomes. This is lethal in humans, but common in plants. The strawberries that we buy in grocery stores are generally octoploid, or 8n. Crossing over and translocation can also seem very similar. However, crossing over occurs between homologous chromosomes, while translocations generally occur between non-homologous chromosomes. Robertsonian translocations can occur between homologous chromosomes. However, they are translocations with breakpoints near or at the centromere, and only on acrocentric chromosomes. And finally, inversion and translocation. These are both structural variations. However, inversion is when a region of one chromosome breaks off, then rotates so that it's upside down, before rejoining the same chromosome. Translocation occurs between two chromosomes, where they swap the regions that have broken off.